right, so what we finished up talking about last Wednesday was external structures, okay? And we said, okay, if you see an organism swimming, what do you know about that? So we talked about, well, we know it's got at least one flagella, but maybe not more than one flagella. We said if it's, bless you, if it's a rod-shaped cell, it means it's a bacillus-shaped cell. So you really want to start teasing apart these types of things when you see it in a question. What do I know? What am I being asked for, okay? Additional things on the outside. So pili are one of these additional appendages. What you will notice is there's kind of two different types. There's pili and then fimbria. Pili proper are really long, hollow components, okay? So what you see, these are typically made of protein, but they're hollow inside. So unlike a flagellum, which is going to be longer, similar in terms of thickness, but longer, this is going to be a solid structure. A pilus, on the other hand, pilus for singular, pili for plural, is going to be hollow in the center. And the reason it's hollow is because it's used for conjugation or a mechanism of DNA transfer. That mechanism of DNA transfer is basically saying, if I want to swap something from one cell to the next, how do I use that? I think we went over this. Why do I feel like we did? So this is when you would basically attach that pilus to another cell, and you'd send something through the pilus over to that other cell. What's commonly transferred is going to be plasmids. And we talked about plasmids. Those are those tiny little pieces of DNA that are independently replicating in an organism. And they can be sent through the pili over to other cells as a mechanism of DNA exchange. It's a mechanism of basically DNA sex, or swapping cells, swapping DNA between one cell and another. We were so close to finishing prokaryotic structure. We've basically been through all of the prokaryotic cell structure components. There's not a lot of them. Remember, there are some that are required, such as a chromosome and ribosomes. You then have additional components that are going to be optional for that organism. Pili are optional. Fimbria are optional. Flagella are optional. We talked about storage granules. Those are optional. So you want to be able to tease apart, what are these cell structures used for, and are they required for the cell, or are they not required? We're going to kind of juxtapose this against some of these eukaryotic cell structures. Now, eukaryotic cell structure, all of this should be review for you. This should all be things that you've covered ad nauseum before. If you don't remember any of your eukaryotic cell structure, you should go back, review this chapter in the book a little bit more in depth, because we're going to talk a lot about this where we distinguish between prokaryotes and eukaryotes. Okay, what do we have? Which one has mitochondria, which one doesn't have mitochondria? Which one has NER, which one doesn't have an ER? You want to be, that's got to kind of be second nature. This is one of those parts where flashcards can be helpful. So you can make flashcards on required cell components, non-required cell components. Or here's what the part is, this is what its function is, what it does. That can be a really helpful study technique when it comes to this kind of components of the course. I like this table. The reason I leave this table in here is because it nicely gives you the difference between the prokaryotic structure and the eukaryotic structure. So we're going to constantly, again, come back to this kind of stuff. Last week we went through and we said, okay, we had a blank table. And we said, what are the characteristics of prokaryotes? What are the characteristics of eukaryotes? All of this, again, is going to keep kind of coming back. When it comes to something like flagella, flagella, for example, can be found in both organisms, and can be found in both organisms for movement. But in a eukaryotic cell, it's got a much thicker arrangement than what it looks like in a prokaryote. Prokaryote being that tiny cell, it can have a small flagellum to use that. But trying to move a much larger eukaryotic cell, it's going to need a much bigger flagellum. This is kind of the difference between, what size motor and propeller you have on a small boat versus something that's got to move something like a cruise ship, okay? There's going to be a difference there in terms of what's needed, even though the structure itself is still the same, and even though its ultimate function is the same. We making sense? So a lot of this stuff are things that we've outlined before, but again, they're going to be things that we keep coming back to. When it comes to those eukaryotes, we talked before, we mentioned this membrane-bound organelles term, and we said prokaryotes are missing membrane-bound organelles. They don't have any of them, but eukaryotes do have them. So what are those organelles? A nucleus is a membrane-bound organelle. If you think of that organelle as being contained or having other parts to it and an inside, these are all going to be membrane-bound organelles. If you've ever heard of your endosymbiotic theory, Endosymbiotic theory states that a eukaryote originally evolved because a prokaryotic structure actually ate smaller prokaryotes. And those smaller prokaryotic cells actually became specialized components in the cell over time. So for example, the nucleus, mitochondria. Mitochondria have their own DNA. Yes, those are the powerhouse of the cell, but we actually believe they came about through this endosymbiotic theory a very long time ago, all right? But all of these different types of organelles are considered membrane bound, and these specialized functions that happen are specialized functions of eukaryotic cells specifically. So you've got your nucleus houses your DNA. In a eukaryote, that's going to be also the site of not only DNA replication, but as well transcription. So transcription happens in the nucleus in a eukaryote. So that concept of taking DNA into RNA. Then you have mitochondria and chloroplasts. Mitochondria are the powerhouse of the cell. Remember, this is where we're going to make all the ATP in the eukaryote. Chloroplasts do the same thing, but they're harvesting that energy from the sun. You have the endoplasmic reticulum, or the ER. This is kind of our packaging section. So remember, the ER has two different components. It has a smooth ER, and then it's got a rough ER. The rough ER is considered rough because it's studded with those ribosomes. And what do the ribosomes do again?
Make protein.
They make proteins, okay? The smooth ER is going to be functioning for lipid synthesis, okay? Synthesis of other macromolecules inside the cell. You have the Golgi. The Golgi is typically connected to your ER. So this little flapjack section here is the Golgi apparatus. And the Golgi apparatus is really like your packaging and shipping component of the cell. So if everything is made in the ER, the Golgi is what decides where does it need to be and how does it need to be to get there. So the Golgi is what packages it and ships it to other components or other parts of that eukaryotic cell. And then lastly, you have lysosomes and peroxisomes. These are internal compartments that are going to hang on to different cellular machinery and cellular compounds. Okay? Membrane-bound structures. Other structures in a eukaryote. The plasma membrane is here. Now remember, in the prokaryote, we talked about the cell membrane. We called it the cytoplasmic membrane. In a eukaryote, it's called the plasma membrane. It is literally the same structure. So how we talked about that phospholipid bilayer before, the same thing applies here. So if you learned it really nicely for a prokaryote, you are good to go when it comes for a eukaryote. All it is another phospholipid bilayer embedded with proteins. It moves according to that fluid mosaic model, so it can expand and it can contract a little bit. It's going to regulate what goes in and what goes out for transport. Okay? It's going to have receptors embedded in the membrane, which are going to function for signaling. A lot of this is going to be very similar to what we talked about for a prokaryotic cell. However, in a eukaryote, they have one additional compound, and that additional compound is going to be a sterol of some sort. So eukaryotic cells are huge compared to those prokaryotes. They're roughly 10 times bigger. When you have a bigger component, you need bigger infrastructure to handle it, okay? So if you think about your house, okay, you've got internal support walls, you have other walls that are more decorative. You understand you can get rid of anything that's not supporting the structure itself, can be moved, can be changed, other things. But those support walls, you're not going to mess with that unless you're putting in headers and beams and all kinds of stuff to maintain that. When it comes to a building like this, you've got a much bigger support, you've got a much bigger framework, okay? Similar to what's the difference between a prokaryote and a eukaryote. Those prokaryotic cells are small. That small nature of that means everything is relatively stable on its own. When it comes to a eukaryotic cell structure, on the other hand, these cells are much bigger and they need some additional framework. One of those components that helps to keep that plasma membrane strong and prevent it from just collapsing on itself and flattening out is going to be these sterols. So all animal cells actually contain cholesterol in their plasma membrane. Okay, you've been taught cholesterol is like the root of all evil. Okay, that's not it at all. All right, cholesterols are actually required for all of your cells to maintain their strength. It maintains plasticity to a certain extent. You know, this goes back to you get old, you get wrinkles, all of this fun kind of stuff happens to you as you age. Part of that is because the sterols in your cells they're not as plastic anymore. They don't move as well anymore. They're not hanging on to as much water as they were beforehand. If it's a fungus, fungal cells all actually contain a different sterol called ergosterol. So ergosterol is in their plasma membrane and function for strength. The difference in the sterols, believe it or not, is actually a target for antifungal medications. So yes, I keep saying this. We're going to go back over and over again to the differences between the different cell types. And this is going to mean a lot for how we study bacterial cells, how we study infections, and how we can treat those infections. A similar thing happens if you get an infection with a fungus. So if you get an infection with a fungus, it's a eukaryotic cell. Your cells are eukaryotic. How we fight those has to be to find a difference between the fungal cell and your cells. One of those big difference is the sterol that's in there. So a lot of those targets are actually targeting ergosterol, even its production, its presence. You can do a lot with targeting that. So think toenail fungus, okay? They have commercials for toenail fungus. You see this little blobby green guy, and he crawls up underneath someone's toenail, okay? It's gross, all right? In the event you get an infection, something like toenail fungus, a fungal infection is hard to treat. This is like 6 months on these antifungal medications because the cell similarity is really spot on. So those drugs have to be given at a fairly low dose, otherwise it harms your cells along with it. So a lot of those antifungal medications typically have a high number of side effects and typically have to be given for a prolonged period of time. They may not ever actually work for you. All right, so if toenail fungus is something that you got, there is a medication that could work. The way the medication is targeting is it's targeting the difference between the sterols that are involved in the plasma membrane of those two cells. We doing okay? It's all coming back to differences in cellular structure. Remember that eukaryote is bigger. It needs more structural framework. So these eukaryotic cells typically have to have bigger structures to support those cells. So a cytoskeleton is one of them, okay? The cytoskeleton is typically made of 3 different types of components. So you've got intermediate filaments, microtubules, and actin filaments. And these guys are actually all strung all along the inside of the eukaryotic cell. It's basically like your internal support walls. A lot of these are going to function, they can function for contraction. They can actually function for transport and movement. So you get compounds that are going to kind of use this as like a little highway system inside the cell. But it's there to basically make sure the cell doesn't collapse on itself. So the cytoskeleton is an important component of eukaryotic cells. Organisms that are living in an aquatic environment or that need to move around can do so by means of flagella. Again, the flagellum that's happening in a eukaryotic cell is really, really big compared to what happens in a prokaryote. In a prokaryote, we talked about how that tail is just a tiny little filament. Here, the tail is actually a whole bunch of microtubules that are arranged in what's known as a 9+2 arrangement. So you've got 9 sets all around the outside, and you've got 2 microtubules that are in the center. And that fat structure there helps to make sure that that eukaryotic cell is capable of moving all around. You can also have cilia. We're going to talk a lot about cilia later, especially when we get to things like respiratory infections and stuff like that. Cilia are like these tiny little hairs that can move things up and around. They can be on the outside of cells to help basically move things around, help them sense the environment, that sort of thing. And then finally, we have that ribosome structure. We've talked about this before. Remember, the ribosome, while is necessary for protein production, is different between prokaryotes and eukaryotes. Prokaryotes have what's called a 70S ribosome. Eukaryotes have what's called an 80S ribosome. Even though the 80S is bigger than 70, number-wise, the prokaryotic ribosome is actually bigger than the eukaryotic ribosome. Okay? So we've got a lot of unique structures in these eukaryotes. What do we think about our cell structure? We took that that prerequisite exam last Monday. Half of that exam was all on cell structure and function. So if you took that exam and you thought, holy cow, that was not good, part of it's coming back to your cell structure, and you want to make sure that you go through it and spend a little bit more time learning it, because it is going to kind of come back.
Did you get them back?
I'm not giving them back. But I will tell you, in this class, the average was a 53%. The highest score on this exam was an 80%. Okay, so, what's that?
Who got that?
I totally don't know.
Do you know what the lowest was?
Not in this class. The lowest in my other class, I had a 27, a 23, and a 17. That was from the other instructor. But they all averaged between 53 and 55%. So the averages of the 5 sections I've looked at here at Dublin were all basically about the same. Basically said that we've got a half understanding of cellular structure and chemistry, okay? So a lot of what I presented was review. A lot of it you were shaking your head like, I know this, I got this, we're good to go. I'm gonna tell you now, just make sure you're really studying that stuff, okay? If you bought the textbook, chapter 2 in the textbook is all on chemistry. It is not part of the required reading for this course because it's technically a prerequisite for this course. If you don't know it, if your chemistry was not good, you took that exam and you were like, what's pH, or how does pH work, or what do you mean hydroxide ions and stuff like that. Go back into that chapter 2 and start looking through that, or go to YouTube or Khan Academy or something and find yourself some videos on atomic structure, the nucleus and what's in the nucleus, protons, neutrons, electrons, understand electron shells, bonding, all of that kind of stuff. We're going to get more into that over the next few chapters, okay? What we're going to transition into now is basically an understanding of microscopy. So, it's clear we're looking at something under the microscope here. What do you think we're viewing here? How many cells do you think we got? 2. Do we see a defined nucleus inside of either of these cells? No. So does that mean you think we're looking at prokaryotic cells or eukaryotic cells? Prokaryotic cells, exactly. So what we have here is actually an electron micrograph. So this is a very high-powered microscope. You are looking at division. So this is cellular division. One cell is dividing into 2. These guys are forming a plate between them, and it's dividing itself out. You cannot see any internal structures because it's a prokaryotic cell and it really doesn't have any, all right? So the more we get into this, the more you start looking under the microscope, the more you're going to become a lot more familiar with what we're looking at, how it works. We are going to spend all lab on Monday is getting used to the compound microscope and how it functions, okay? We basically have an hour and a half to look at some samples, look at a bunch of different slides. We're going to start with a slide that's got the letter E on it. It's got a little piece of E newsprint because you know what the letter E looks like, so you can find it, you can know when it's in focus. And then we'll start branching out into different types of prokaryotic cells to see if you can identify them. What we are using is going to be the light microscope. Okay, so for this guy, once you practice working with this, once you get used to this, all other microscopes are very similar in terms of how they work. In general, what do we have going on here? So we have the light is always going to be shining up from the bottom in a compound microscope. The light goes up through a condenser. The condenser focuses the light. It then goes up through the stage. On the stage, you're going to have a slide sitting with the specimen on it. So we want the light to come up through that slide. It then goes up through what's known as the rotating nose piece, which is carrying the objective lenses. This has got 4 different lenses on it. Your microscopic class also has 4 lenses on it. We will always start with a lower objective and work our way up to a high-powered objective. But this has got 1 set of magnification here. From here, it goes up through the oculars. The oculars as well are going to add to the magnification. Typically, most microscopes have a 10x magnification going on with the oculars. Your microscope is the exact same with that. So in general, we are limited in this case. The highest objective that we have is going to be a 100x objective. So our total magnification is approximately 1000x. Most compound microscopes are limited to 1000x magnification. You can get higher power objectives. It just depends on how much you're willing to spend. Okay, the microscopes we have in class are $1,800. Okay, each. Yeah.
On the pre-lab, it was like if you add 40x to it, what would it be? Will you explain how that works?
Yes, and then we're going to get to that in another slide or two.
Okay.
It's you got to multiply them together.
Oh, okay.
So if you're trying to calculate total magnification, you've got magnification from this one and magnification with this. You basically multiply them together, so you just multiply anything by 10. So if you've got the 40x objective, You've also got 10x from the oculars. 40 times 10 gives you a 400x total magnification. So this is a basic light microscope. It's also known as a compound microscope. Okay? You've got a lot of other types of microscopes that can go along with this. So an electron microscope can magnify images in excess of 100,000x. So you're limited to 1,000x with what we have in class. You can go 100,000 times that when you use an electron microscope. You have scanning probe microscopes. You've got all kinds of different microscopes that basically get finer and finer in terms of their level of detail and what you're able to actually see. The whole point of this microscopy is that light's going to pass through the specimen and then through magnifying glasses, and then you see it, okay? That bright field microscope we've got in class is the easiest to use. It's the most common form of a microscope that's going to be out there, right? There are three main components to microscopy, and you want to understand how these work and be able to work through them, okay? The first one is going to be magnification. Magnification is amplifying your image. So this is making that image larger to see. We're looking at these prokaryotic cells. These prokaryotic cells are too small to be seen when single-celled with the unaided eye. Therefore, if you're going to see an individual prokaryotic cell, you've got to be looking at it through the microscope. So magnification is a big component of this. But you understand it's not just about how big you make something, okay? Because there is also what's known as resolution. I like to give examples of, your digital camera. Nobody uses a digital camera anymore. They just use a camera on their phone, okay? You've all done this. You zoom in on something, and then what happens? It's blurry, okay? It gets blurry because your magnification and your resolution are not basically lining up anymore, okay? Same thing happens with a microscope. In order to see an image clearly, it's not just about the magnification, it's also about the resolution that goes with it. In addition to these two components, you also have what's known as contrast, a difference in color. You saw this today with the dissecting scope. Most of what you were looking at on that plate was white, off-white, cream, pale yellow. You noticed that the yellow ones were much easier to see than those other ones that were just more white against the plate and all you saw was a little bit of an outline. So contrast is referring to the number of visible differences in terms of color that you're seeing under the microscope. The more color we can bring in, the easier it is to actually view something under the microscope. So today's use of the dissecting scope, what you had was a little bit difficult to see. Unless you picked up one of those like flower samples or something like that that was over to the side. Some of those mold samples were different as well. What we're going to see on Monday is we're going to play around with this a little bit. We're going to look at that letter E. There's a lot of contrast there. Black E against a white background. Good contrast. We're going to have a lot of slides that are already stained. So we've got some that are purple and blue and green. And you're going to see how those cells really stand out. They're easy to find, easy to focus on, no problems. And then the last part of that lab on Monday is going to be looking at a pond water sample. What's in the pond water? Doesn't have a lot of contrast. These organisms are pale green, are pale yellow. So it's hard to find them against a white background. So all three of these components work together in microscopy to help you see an image clearly and to help you be able to focus on something. All right, so individual parts, okay? So remember, your microscope's got two magnifying lenses. That's what makes it a compound microscope. When we talked last week, we said Leeuwenhoek created the first microscope, and his microscope was a single, simple microscope. It was simple because it only had one set of lenses. These microscopes all have two sets of lenses, which makes it a compound microscope. We've got the ocular component, and we've got the objective component. Both parts of those have magnification to them, and they combine to enlarge objects. In order to calculate the total magnification, you have to multiply them by each other. You will notice every time you see this, you've got to write the X on the end, otherwise the number is totally meaningless in every way. So if you've got a 10X ocular piece, and you've got the 100X objective lens in place, the total magnification you are looking at is 1000X magnification. Resolution, that other component. Resolution is technically defined as your ability to see distinct objects next to one another and see them as distinct. What you can see here looking at similar cells here, we've got a cell and we've got some other air pockets around it. These guys, in a light microscope, even at 450x magnification, this looks blurry. These guys look on top of this other cell. You can't identify them separate from one another. But when you suddenly have a better microscope that's got better resolution, resolving power resolution, same thing, okay? Even at the same magnification, now we can see that there's actually a distance here. Now we can see them apart. Now we can really tell this is the nucleus of that cell. We can see the cellular boundaries much more evenly. There is no difference here in magnification. The difference is going to actually be the resolution, okay? And this is what you're looking at where you then back off on the zoom until it becomes a little bit more clear for you to see. So it's a difference between seeing something really nice and crisp versus seeing something that looks like a blob. Here's another way of looking at that. It's finer points of details. The higher the resolution, the higher the quality, the more fine points of details you can see. So something that's got higher resolution, looking at something like your hand, you're taking lots of different fine points of detail. You're able to clearly see your hand in that outline. Something that's got low resolution is going to look really blurry and not very nice, okay? This is like my kids got a digital camera. It was like a $20 digital camera that my mother-in-law bought. Every picture on it is blurry. Why is it blurry? Because it's got no resolution, okay? Yeah, sure, they can zoom in, and they love to zoom in, see everything, okay? It's a cheap camera. It doesn't have good resolution to it. That's why everything looks blurry, all right? Back in the day when you had to buy a digital camera, you'd look at megapixels and all of these different components, okay? We don't have that anymore, unfortunately. You just get a camera on your phone. Going with resolution, when it comes to dealing with the microscope, when we work with this microscope, we've got one lens on that microscope that's known as the oil immersion lens. That's the highest lens on the microscope. It's our 100x objective. That 100x objective only works if you use immersion oil with it. And the reason here has to deal with light and refraction of light. So when the light hits a different substance, light is actually going to bend. So in this case, if you're looking at this cup of water with the pencil in it, you see it almost looks like the pencil is broken between what's down in the water and what's hitting the air. That's because the light is actually bending and refracting differently between those two different components, okay? Same thing happens when you use a microscope. Any light that shines up, depending on how it hits and the refractive qualities here, you're going to lose light out the side of the microscope. It means things are going to be blurry. You're not going to be able to see it clearly. With this 100x objective lens, it's got a very small difference in distancing gap here and aperture. So because of that small difference, we add in this immersion oil. The immersion oil has the same optical properties as glass, so everything looks the same, and now it forces all of the light up through the objective lens. If you do not use the immersion oil with the 100x lens, you're not going to see it clearly. It's always going to be a blur. So you've got to make sure that that's working correctly, and we will practice this on Monday to get it to work right. But everything is coming back to that resolution and that refractive index.
How much immersion oil do you put on this light?
We're going to put one drop. So you're going to put just a drop right where you see the light coming up, and then you will actually swing this objective lens into place. When you move the objective lens over that one drop of oil, it's going to gather it up. It's just going to swoop and kind of bridge this and stay here. If it's not in focus to start with, the oil won't touch, and then you know, wait a minute, whatever I was looking at before wasn't quite right. So you just need one drop, and as long as it was in focus, this will swing in, and it'll be totally good to go. How we doing? Are you okay? Yes? No? Yeah? Okay. Contrast is the last component. So this is going back to the number of visible shades. Higher contrast, in other words, more color, is much more useful for staining, okay? If we stain cells, we impart a color on them, okay? Can you think of a situation where you would not want to apply a dye to a cell? You want to see it alive, exactly, all right? So when you impart a stain on a cell, you kill it. So You kill it, it's dead. You can't see movement, it's not gonna grow anymore, you won't watch it divide. That's it, that's the end. If you wanna see a living cell and you wanna watch it divide or watch it move around or see how it changes over time, staining is going to kill it, okay? So we're gonna look at this on Monday. We're gonna look at some cells that are live under the microscope. We're gonna be able to watch these tiny little prokaryotes moving all around, skipping all around, bless you. You can see their flagellum. They're cool. If we would have stained those, we would have had an easier time seeing them, but we would have killed them, and then we never would have been able to see them moving all around. So how we choose to work with the microscope and what we choose to do, a lot of it is going to depend on our ultimate needs. If you want to keep a cell alive, you're not staining it through a typical method. If you don't care if it's alive or dead, you just want to observe it, staining is going to impart a contrast and it's going to make it a lot easier for you to look at it under the microscope. There are lots and lots and lots of different types of microscopes that are all meant to increase contrast and make it easier for you to see. We are not going to work with any of these other types of microscopes in class. But depending on what your ultimate needs are, you can vary these and use them. So in my previous life of research, I used all of these different types of microscopes. It really just depended on what I was doing. I used a lot of fluorescence microscopy, where we would basically tag these with a UV wavelength, UV chromophore basically. And you'd hit them with different wavelengths of UV light and they would just pop out it, okay? I've also used a confocal scanning laser microscope, okay? This is actually able to take 3D sections through a slide and build a 3D image of the cell over time. A fluorescence microscope is gonna cost you about $20,000. A confocal scanning laser microscope is about 250 grand, okay? It's got to be on an anti-vibrational table. All of this stuff has to be just right because you've actually got a laser coming at it from the side. You've got the microscope, it's attached to a computer. All of that alignment has to be totally right, otherwise it doesn't work. So examples of applying contrast. So basic bright field microscope, looking at a paramecium, you see this one at the top. This is what you could see on Monday under the microscope. This is actually a little bit more of a magnification than we will be able to see, so it's a little bit larger than what you're going to look at. But that's a general paramecium, it hasn't been stained. Taking that same paramecium and putting it under a dark field microscope. So bright field has the background lit, dark field, the background is black or dark, and then you're looking at differences within this. This does allow you to see a little bit better in terms of contrast, it outlines it a little bit better. All you've done between these two microscopes is change the light, change the condenser that's associated with it. The last one is a phase contrast microscope. This one is really cool in terms of imparting contrast and allowing you to really see distinguishing features. So just changing that contrast allows you to see a lot in terms of internal and external structures. Because now what we're looking at, we can clearly see the nucleus. You can see the genetic information in the nucleus. You can see the outline of the cell. You can actually see that this guy is covered in all kinds of tiny little flagella that are going to help for its movement. You get none of that by looking at it under the bright field microscope, okay? So imparting a contrast is really important for seeing cells. Monday's exercise is going to be the only one we do all semester where you're looking at live cells. Everything else from this point forward, we're going to be imparting a contrast. We're going to be performing staining to do that, so that we clearly see the cells themselves. Here's a look at fluorescence microscopy. So it's tagging it with a fluorescent dye and then hitting it with specific UV wavelength light. And you can see they light up. It's easy to tell where the cells are because they're basically fluorescing on your screen. That confocal microscopy, again, really cool because you can end up with a 3D image. So what this does is it actually sends a laser beam through sections on the slide. Super, super thin sections where it's passing the light through. Each individual section on that slide is going to give you a colored output. So what you see here, red is outlining the cell, and green is actually anything internal to the cell. So you can see cellular boundaries here and cellular components. When it builds back that 3D image, everything on the outside of the cell is red, so that's why this particular image looks red and looks much more like a blob because it's filling out that 3D image of the cell. There's a lot that you can do with a microscope. It's all about having the right type of microscope that you need with the right condenser, the right components. Depends on what your needs are, depends on what cells you're looking at. Because if you want to see something super small, think virus, what you have to have is going to be an electron microscope. An electron microscope is different because in this case, instead of hitting it with light, you are actually hitting it with electrons. So you're bombarding that particular component with electrons. This does have objective lenses and condenser lenses, so we are getting some of that. It's flipping the image to a certain extent, but now we've drastically increased the resolution as well as the magnification. How to work this, it actually requires that everything is in a vacuum. An electron microscope is going to cost you about $2 million, okay? We do not own one of those at Columbus State. Most larger institutions only have one or two on site that they use, and they obviously don't just let anybody walk up and use the electron microscope. You've got to give them your specimen, they look at it, and then they give you the results back. So you really, this is not one you're playing around with. You have to have a significant need to be able to use the electron microscope. But if you ever want to take an image of a virus, they're so small, no traditional microscope is going to get you that type of amplification. You've got to look at an electron microscope in order to see those. What do we think?
Have you ever gotten to use an electron microscope?
I've seen one once. I gave them a sample and got an image back. That was the closest I got to it.
And are these all like purchased through research funding?
Yeah.
Yeah.
Grant funding.
Yeah.
Yeah. And typically when it comes to something like an electron microscope, that's like multiple grants come together to buy one, and then the college has to pitch in money too, and stuff like that. So, yeah. The confocal microscope was individually grant funded, and that was like a huge deal. It was like, oh, this guy's got enough money on this grant, we bought this $250,000 microscope. That was fun to play with, but yeah.
So whoever came up with these microscopes is living a nice life.
Is living a nice life, right. Even the companies that make them are living a pretty, yeah. So if we're gonna look at these cells under the microscope, we've gotta stain them, all right? So we are gonna do a lot of different staining techniques. Exercises 3, 4, 5 in the lab are all on different staining techniques. We're gonna then come back to those multiple times during the semester to make sure that we're seeing these cells appropriately under the microscope. So you're gonna get a lot of practice with this kind of stuff over the next 2 weeks. We call them stains, we also call them dyes. They mean the same thing. Basically what it is an organic salt, and it is going to carry a charge, and that charge binds to cellular structures and gives them a color. All of this is coming back to charge, okay? So if it's a basic dye is going to carry a positive charge, and it binds to structures that carry a negative charge. If it's an acidic dyes carry a negative charge, and they bind to structures that have a positive charge. What you want to think here is opposites attract. Whatever your cell structure charge is, you want to come in with the opposite charge on the dye in order to actually be able to stain it. Last week, so last Wednesday, we talked about how the cell wall has lots of negative charges. We talked about gram-positive cells have teichoic acid, and that teichoic acid has a negative charge to it. Other components of the outer membrane also carry negative charges. So prokaryotic cells have lots and lots and lots of negative charges. When we want to stain our cells in class, we are using lots of basic dyes because the basic dyes are positively charged and they stain those negative cell structures. In the event we were to take your prokaryotic cells and we were to stain them with an acidic dye, typically what happens is that they repel one another and it stains the background, okay? This is just like those little magnets with the little north and south ends you played with as a kid. The north and south will attract one another. Two ends of the same charge repel one another, okay? Same thing happens here. If you come in with a negatively charged dye against a negatively charged cellular component, those dyes are typically opposite of one another. Interestingly enough, most prokaryotes have different cellular charges than human cells. Human cells have lots and lots and lots of positive charges, and therefore commonly what we use to stain human cell samples is actually going to be a whole lot of acidic dyes, okay? So the dyes we use in this class are going to be different than the dyes that are used to stain slides for anatomy or something else, okay? We talked about charge. We're also going to include these terms positive staining and negative staining, okay? So positive staining is when the cell itself takes up the dye. What you see in this first one is that you have a basic dye, so it's got a positive charge. That positive charge is attracted to negatively charged cell components, and this cell in this case all turns blue. Here, opposite. We are coming in with a negatively charged dye. It's going to get taken up by positively charged components of the cell, and now this guy is going to turn pink. Methylene blue is a common stain for prokaryotes. Eosin is a really common stain for human cells. The most common staining procedure in looking at human tissue is called an H&E stain. E stands for eosin. It's adding this particular acidic dye to it, okay? Either way, these are known as positive staining because the cell itself is taking the dye. Negative staining is where you get that repelling. Same charge on the dye to the cell structure, and now they're repelling one another. In this case, it's going to turn the background a dark color compared to the cell. So what have we talked about here? Okay, we've hit a lot of positives and negatives in the last week, all right? We talked gram-positive. Anybody remember anything about those gram-positive cells? Exactly. These guys have a thick peptidoglycan layer in the cell wall. We talked gram-negative. Conversely, these guys have a thin peptidoglycan layer plus an outer membrane. We talked positive charge and negative charge. Now we're also hitting up positive staining which means we stain the cell, and negative staining which means we stain the background. This is another one of those places where some flashcards could become really handy, okay? You've got to be able to tell these guys apart. You never want to hear positive or negative in this class, because it's what positive or negative what? You're talking about positive charge, talking about gram-positive. What are you dealing with? Each one of those terms is telling you different information about the cell. Because you could have a gram-positive cell that is negatively charged. Because it's negatively charged, we have to come in with the positive staining and basic dye, positively charged dye, in order to stain it. We've just combined positives and negatives all in the same thing to talk about staining one particular cell type. Okay? So you want to make sure that you can keep them all straight. Okay? If it's a basic dye, it's positively charged. If it's an acidic dye, it's negatively charged. What do we think? I'm going to try not to get confused. We'll talk about this a lot. We're going to spend a lot of time staining in class. The biggest thing that I can say as we work through these procedures is know that we're going to use basic dyes a lot. Okay? Even though we have gram positives and gram negatives, both of these contain cells that are negatively charged. Okay? The gram positive or negative has nothing to do with the charge on the cell. It's totally separate. So we're going to spend a lot of time doing using basic dyes for positive staining. We are going to do one type of negative staining in this class. We're going to look at a capsule. The capsule stain is a negative staining process because we're going to outline the capsule. We're not actually going to see it clearly. Okay? How do these dyes and staining procedures work? It's all a series of steps. None of the steps are complicated. It's just about watching your timing and making sure that you're getting everything on there. So again, we're going to use lots of basic dyes in microbiology. They're much more common than acidic dyes. Common basic dyes: methylene blue, crystal violet, safran, and malachite green. We are using all 4 of those dyes within the next 2 weeks in this class. Okay? In general, how do you do it? So the first thing that you're going to do is you're going to get your cells and you're going to spread a thin film of them over the slide. Most of the time, we are going to be taking cells from a solid surface. So we'll actually spread these dyes in a drop of water to make sure that we get an even and thin film across the slide itself. You want a solid, thin layer of cells. If you've got cells that are all clumped, they're not going to stain evenly. Okay? We're then going to allow this to dry. After it's dry, because there's typically water involved with this, you're going to pass the slide through the flame. We're actually going to pass the slide through the flame 3 times. This process is called heat fixing, and that process actually helps to change the membrane structure of the cells. They actually, they become easier to take up the stain, and they actually melt just ever so slightly onto the slide so that they stick there a lot better. If your slide is wet when you do this, you boil your bacteria, and then that's not going to work. Okay, so we're going to make sure it's dry, then we're going to heat fix. Then we're going to add in our dye, and we're going to let it sit on here for a set amount of time. Some of the dyes we work with are going to be 1 minute, 2 minutes. We will work with some that's going to say you got to leave this on there for 15 minutes before it actually stains anything. Okay, so the timing is going to vary. You will rinse it with water, and you will blot it dry a little bit. Then you stick it under the microscope and you look at it. Cells are all then going to have taken up the color. This is the general process for everything that we're going to use in class. Get it on the slide, let it dry, heat fix it, apply your dyes, and then check it out under the microscope. We are going to do 2 different types of staining. We're going to do simple stain. This is first next week. And then we are going to do a basic differential stain the following week. Okay? So a simple stain uses one particular type of dye, and in this case we're using a basic stain because we're staining prokaryotic cells. Everything takes on the same color. So just like in this previous slide, we stained with one type of dye, everything is going to look blue. Okay? So everything turns the same color when you perform a simple staining procedure. There's no differentiation between cell types. You can see different sizes under the microscope or different shapes, but everything is going to be the same color. On the other hand, you could use what's called a differential stain. Differential stains are going to use 2 or more types of dye, and it's going to allow you to stain different cellular structures, different colors. Okay? The 2 most common ones in microbiology are 2 that we are going to perform in the lab, and that's going to be a gram stain and an acid-fast stain. Gram stain, you get it, is going to be capable of showing us the difference between gram-positive and gram-negative. So for this gram stain, was developed by a guy by the name of Hans Christian Gram. Hans Christian Gram really just wanted to see cells under the microscope. We actually did not know about this difference in cell structure when he was creating the staining process. He kept doing these stains and applying these dyes thinking everything should look the same, and what he kept finding was, wait a minute, I'm getting 2 different colors. Why would I get 2 different colors? What's happening here? And it was then that he started to think, wait a minute, I get 2 different colors because they have different cellular structures. Okay? So the bacteria can actually be separated into whether they are gram-positive and have a thick peptidoglycan layer, or whether they are gram-negative and have a thin peptidoglycan layer and an outer membrane, if you perform this particular staining procedure correctly. So gram-positives are always going to stain purple. Gram-negatives are always going to stain pink. And what you're looking at on this slide is that you can see we've got individual purple coccus-shaped cells. So coccus-shaped means it's got a circular shape. And we see pink rod-shaped cells or bacillus-shaped cells. The pink ones are the gram-negatives. The purple ones are the gram-positives. How do we do a differential staining procedure? Well, this means we've got to apply those stains in a very set series of steps in order to get the colors to look right in the end. So we're going to use what's a 4-step procedure. We've got a primary stain, which is going to stain all the cells. So in this case, we're using crystal violet. Everything in here is going to stain purple, regardless of what it is. The next thing you have is what's called a mordant. A mordant is cool because it actually helps to seal in the primary stain. It complexes with the crystal violet. Crystal violet is really more of a bluish color. The iodine is more brown. When the iodine hits that crystal violet, it binds to it and it creates this larger complex that seals it inside the cell. That blue and brown combo really takes this to more of a purple color when you're done with that mordant. You will then come in with a decolorizer. In this case, the decolorizer we're going to use is a combination of acetone and alcohol. That acetone alcohol is interesting because it removes the primary dye from the gram-negative cell only. Ultimately, what that decolorizer is doing is it's actually dissolving the outer membrane. It says, listen, outer membrane, I'm just going to poke a whole bunch of holes in you. And it pokes all of these holes and it releases the primary dye. So at the end of step 3, your gram-positive cells are still purple because they have that thick peptidoglycan layer that's still retaining the primary dye. The gram-negatives, on the other hand, are going to be clear, colorless, because those gram-negative cells have released their outer membrane. The outer membrane was that outermost structure that was holding in the dye. So that these cells then become clear or colorless. And finally, you come in with a counter stain. In this case, we're coming in with safranin. That safranin now binds to the exposed peptidoglycan layer underneath. What that means is that the gram-negative cells appear red or pink. The gram-positive cells are going to stay purple in color. Those gram-positives do take up some amount of that pink color, but because the purple is darker than the pink, the color really doesn't change here, okay? It's dependent upon the charge, and it's dependent upon the color, okay? So the order of the reagents is actually really, really important in this particular scenario. In the event, for example, you forget to add your alcohol. If you forget to add your alcohol, these guys never go clear, which means they can't be pink in the end. Means everything still stays purple. If you forget the iodine, you're going to be more likely to decolorize your gram-positive cells, and you're more likely to end up with everything that's pink in the end. If you switch the order and you add safranin first and crystal violet last, your color is not going to be the same. The charge on both of those compounds is exactly the same, so it's dependent upon the structure. So if you added safranin first, all the cells turn pink. If everything is pink and you add a brown colored dye, what do you think that does? You think they're a nice pink anymore? No, they're like a pinky brown color. They're a dark color now. When you come in with the alcohol removes the outer membrane. You're still removing the outer membrane. So gram-negative cells become colorless, but these gram-positives are this pinky brown color. And then if you came in with the crystal violet last, it's a blue color. It's still going into these different cells. Gram-negative cells would take on that dye and be really nice and blue. Gram-positives are going to take it up a little bit, and now they're pink, brown, blue, kind of all mixed together. They're going to look very similar to the ones next door. So these colors and the steps, everything has to be combined in exactly the right way. Otherwise, you don't get the desired result you're wishing for. Because it all comes back to what am I doing to the cellular structure and what charges am I dealing with. Both of these dyes have the same charge. So it's really an understanding of where is my cellular structure altering, what's going on with the particular reagent, what's its purpose. Mordant is going to seal in whatever it is, regardless of what dye you put up here. If it's a pink dye, it's bind into a pink dye. If it's a blue dye, it's bind into a blue dye. Okay? So when we do the gram stain, the order that we do it in is going to be really important. Another staining procedure that's also differential is going to be an acid-fast stain. This is going to be used to stain organisms that resist conventional staining. In other words, they have lots of waxes and lipids in their cell walls, and they don't evenly take up the stain. An example, Mycobacterium tuberculosis, okay? You've all heard of TB, okay? If a patient has TB, that's a hard to treat disease. It's in their lungs. It's a good 6-month antibiotic protocol to try and get rid of it. It's a multi-drug therapy. Why? Because Mycobacterium tuberculosis has what's called an acid-fast cell wall. It's got all of these waxes and lipids in the cell wall that really resist staining. They also resist drying out as well. So when we try to stain Mycobacterium, we can't just use a gram stain procedure to do it because its cell wall is not the traditional gram positive or gram negative. So in this case, this acid-fast stain, it actually uses heat to drive in the stain. These cells are going to actually turn a red color. And then we come in and we stain the background with a blue color, okay? How do we normally see if someone has TB or not? We want to do the staining. So we can do two things. We can do a TB test. If they want to see if you, so if you do a TB test, a lot of you have had this, okay? They scratch you or they inject you underneath with a small amount of the organism's antigens, so the foreign particles. If you have a reaction to it, within 48 to 72 hours, you're going to start to swell. You're going to get what's called a wheal and flare reaction. It's going to get red, bless you, and it's going to start to kind of expand on you. If you have that reaction, it does not mean you have an active TB infection. It simply means you've been exposed to that organism at some point in time. That could mean you had a patient years ago who had an active infection, you got exposed to it, but you never got true-blown infected. Your body made an immune response anyway. If you got the vaccine, you would test positive on a TB test. We do not give the vaccine here in the United States. Other parts of the world give you a Mycobacterium tuberculosis vaccine. You still test positive. Or you could test positive because you actually have this infection going on. To confirm that infection, commonly what they'll do is a chest x-ray next. They're going to see if you have all of these infiltrates in your lungs that are what's going to happen with this infection. The other thing that they can do, they can have you cough up a whole bunch of schmutz from your lungs. So yeah, they need you to produce some sputum, okay? So that's a nice deep cough, and that's hawking a loogie right into some container, okay? And then they're going to do this particular stain. When you do this stain, most of what you cough up is your cells and other schmutz from your lungs, okay? And it's all going to turn blue in the background. But if you have TB, active TB, you're going to be able to see these particular guys all come out with this particular stain. These will actually turn more of a red color. It's kind of hard to see on this particular, with the camera here. But they're turned more of a red color. So this is another way of diagnosing someone with an actual TB infection. It's not enough to just have the test because there are a large variety of possibilities that would give you a positive reaction. Even if you had TB 10 years ago, you would still test positive on that TB test today because you have antibodies against it. But this is a stain. We are not actually doing this stain in class because Mycobacterium tuberculosis is terribly nasty, contagious. It's a higher biosafety level. So we're not playing with this particular culture in class, but we are going to show you some slides that have already been stained. They were basically taken from patients with active TB infections. They had them cough up some stuff, and then they stained it. So you will actually be able to see this, a similar looking slide in class. We've got some that are whole mounts as well that was just pure culture of that organism that you'll be able to see.
Why don't we vaccine in the U.S. for TB?
In the United States, it's more of a numbers game, believe it or not. It's more like, okay, how much is it going to cost to vaccinate all of these people? When we test you all, we use that TB test. If we've given everybody the vaccine, the TB test doesn't work, and then we have to give everybody a chest x-ray and other stuff. So it's trying to save money on one end, but at the same time, if we have a massive outbreak of TB around here and nobody's vaccinated, that becomes a problem. Yeah. We have less of an issue here with that particular infection, but I would not be surprised if that changes over time. Yeah. And if you're traveling to different parts of the world, they would say, okay, look, you know, we don't give you the vaccine here, but we're recommending you get it due to where you're traveling to. And then every point after that, when you have to have the test done, you're gonna test positive. So, you'll have to prove, I had the vaccine, here's my papers. They may give you a chest x-ray anyway. It's more about money than it is anything else.
Won't they do your quantiferin or won't they test it other
They
ways?
can test immunoglobulins to see what the level is there.
And then after that, they'll kind of go.
Yeah. Depending, so if you've got the vaccine, or if you get it on your own, you make specific immunoglobulins first, and then less ones longer term. So they can actually test the amount of immunoglobulins you have, your antibody titer, and that can determine either how long ago you had the vaccine or how long ago you had the infection. Yeah.
Wait, so if you wanted to, you could ask for the vaccine here?
You could, they might not, it's hit or miss as to whether or not they carry it and they have it, or they can get it for you. Or if your insurance will pay for it, or how much it's gonna cost and everything else. Yeah, they do have it around in this country, it's just they're not commonly giving it. Yeah. You can do all kinds of staining, guys. It's all about what you wanna see and understanding charge. So we are gonna do the capsule staining class. This is a negative stain. What you're looking at here, we've actually stained the background kind of a dark color. The capsule is actually this clear area around it, and you can see there's actually 2 cells in the center of this. So we will do this negative staining procedure later on to help you see the capsule and just how thick it is around some of these cells. When we do it, we're actually going to stain the cells in the center pink so that you can see the small part of the cell and how thick that capsule gets around it. We are going to do an endospore staining class. So this stains that endospore structure that we learned about last Wednesday, so that dormant cell structure. The endospore itself stains green, depending on which type of dye you choose to do this with. And then the vegetative cells, or the living cells that are left, are still going to stain a pink color, but you'll see 2 different colors under the microscope. You can also as well do a flagella stain. This imparts, it's an interesting component to the dye that actually thickens up the flagella and makes it more easy for you to see. Under our normal staining procedures, you're never going to be able to see the flagellum in class. It's just way too thin and too small. This particular staining procedure actually causes the dye to stick to it and fatten it up, so it makes it easier to look at under the microscope. We do have some pre-done slides of this. We are going to look at this as well. So over the next couple of weeks, you're going to do a simple stain where we just add 1 dye and see what it looks like. We're going to do a gram stain where we can see gram positive versus gram negative. You are going to do a capsule stain, an endospore stain, and you're going to do another one. We're going to observe the acid-fast cells. We're also going to observe flagella stains as well. Okay, so we're going to get a lot of microscope time coming in over the next week. When we're looking under the microscope, we want to help start to focus on the cells themselves and know what we're looking at. So again, you want to use your terms. Flashcards can be a big help with the terms. If you see the word coccus, that means it's a round shape. So it looks like a little sphere. For example, Staphylococcus aureus. Coccus is in the name of that genus, okay? That is a round shaped cell. Staph aureus, when you do the gram stain, looks like little bunches of grapes. Looks like purple little grapes all under the microscope. I will be honest, we're normally here 10:30 to 1:30. I'm hungry. I don't know about you, okay? I start to revert to everything as food. Everything starts to look like food under the microscope. These guys tend to look like little bunches of grapes under the microscope. A bacillus shape is more of a rod shape or a cylinder shape. Not to be confused with the genus Bacillus, although if it's a genus Bacillus, which will be in italics, all of those are typically bacillus shaped as well. But it's referring to a rod shape. These can be a teeny tiny rod. They can be a really long rod. If it looks like a cylinder, it's going to take on a bacillus shape. If it's a round shape, it's going to be a coccus shape. Other types that you may see. Coccobacillus is technically a short round rod. This is where the food comes in, because I will say this looks like a tater tot. Okay? It's not quite round. It's not quite a rod. It kind of looks just short and squat in between. Okay? So Coccobacillus to me looks like little tater tots under the microscope. We will see lots of little Coccobacillus or guys under the microscope. Vibrio. Vibrio is actually a curved rod. Keep in mind that it depends on the plane that you're viewing it in. So if you're looking through the microscope and the curve is headed towards the slide or something, it's not going to look curved to you. It's got to be sideways in order for you to actually see it. But Vibrio shape is a curved shape. You have spirillum and you have spirochetes. Both of these are more of a corkscrew or helical shape. The spirochete is much more corkscrew-like. The spirillum is a little bit more relaxed on that. These are much less common. Coccus and bacillus are the two most common shaped organisms that we're going to see, but there are times where you're going to see some other ones in this class. The other thing that you want to know is you're going to see these cells and they're going to be grouped together. This is where we go from those nice textbook examples to I need to help you understand groupings, and so I've drawn it myself in PowerPoint, so I apologize, okay? As cells divide, they go through binary fission, they typically stick to one another, and they can divide and stick on any plane. So what you're looking at the top is two bacillus-shaped cells that are stuck together end to end. You can have bacillus-shaped cells that stick together on their long plane as well. You can have coccus-shaped cells that all group together in one way or another, okay? So they are going to stick together depending on their plane of division. If you see, for example, so coccus is referring to the shape, and then you can start to add in extra terms. So diplococcus is referring to two coccus-shaped cells that are always next to one another. For example, Neisseria gonorrhea always is associating in this two coccus-shaped cells next to one another. If you work in an STD clinic and you're taking scrapings from a patient, the only disease you would ever find in that particular environment that's going to look like this means that patient has to have Neisseria gonorrhea. It can be a very quick diagnosis and they do it commonly, okay, is to basically take a scraping, stick it under the microscope and see what you've got. By looking at general cell shape, they can determine a lot of different types of bacterial or protozoal STDs that can be found, okay, very quickly. Strepto means chains. So streptococcus is chains of coccus-shaped cells. So looking at them all on end to one another, that's a streptococcus conformation. So we're going to have Streptococcus lactis in this class. It's going to look like chains of these coccus-shaped cells all strung along with one another. You can get packets, you can get clusters, they pretty much look the same, okay? So clusters is when they divide along random planes. So staphylococcus, staphylo is meaning cluster. In this class, we're going to spend a lot of time with staph, and we're going to spend a lot of time with strep. I will tell you, it is very hard to see a bunch of chains that are all on top of one another and not have them look like clusters, okay? So take some of these groupings with a little bit of a grain of salt, because a lot of it is going to depend on what species you're working with, how old those cells were, and how much you broke them apart when you used your loop to get them onto the slide. But it's all going to be about groupings, okay? So we finished basically on time, which is good to go. What I want to do at the moment is to talk a little bit about study components, okay? So we've got 2 more chapters to go before we have our first exam. We're going to get through the next chapter, which is prokaryotic growth. We're also going to talk about viruses before that exam. Next week, we're going to talk about your first homework assignment. Next week, you will also have, we got to get through Monday, so we're doing exercise 2 on Monday, but we'll have pre-lab 3 due next Wednesday, so make sure you're staying on top of that. When it comes to studying for this class, don't wait until right before the exam. So we covered a lot in terms of cellular structure. Half of this is supposed to be review, and half of this is going to be new. I think what I've seen is that it's not all going to be reviewed. Make some flashcards with cell structure. Go back to that outline. Start filling that in. You can already bring up the study guide and start working your way through it. If you know now that you're going to need some help or you're not understanding some of these terms, spend 10 minutes, make some flashcards, and focus on those, going through the different types of staining or positive, and where do I see positive and negative, how all of that works. It's only going to benefit you from spending a little bit more time now working with the material than leaving it all at the very end, okay? All right, we are good to go for today, guys. Have a lovely weekend, and I will see you